Today we're going to talk about an approach to T-cell lymphomas, and the topic of my talk today is assessing the knowledge gap. And many of you may be expecting the kind of talk that I usually give, talking about the disease and how we treat it, et cetera. But actually, I thought that today I'd take a little bit of a different approach and really talk about what we actually know about this disease and really try to incorporate some of the science into the talk as well. So in terms of disclosure, although I do participate as a consultant for a number of companies, I'm actually not talking about any drugs today, so there's no conflict of interest. So focusing on T-cell lymphomas are rare, but the interesting thing about T-cell lymphomas is that they actually are as common as Hodgkin's disease. There are about 66,000 cases in the United States per year. And you can see the distribution with respect to other kinds of B-cell lymphomas. T-cell lymphomas comprise a small number of lymphomas. 10% of all lymphomas are T-cell lymphomas. There are 6,600 cases of T-cell lymphoma in the U.S., and there are actually about 8,500 cases of Hodgkin's disease, so very similar numbers. However, if you do a Medline search, you'll see that there are over 6,000 papers related to Hodgkin's disease and 1,300 related to treatment of Hodgkin's, whereas for patients with peripheral T-cell lymphomas, there are only 524 papers and only 82 of them address treatment. Therein lies the lack of evidence base for the management of this disease. So if you look at the overall survival of lymphomas in general, you can see that these are predominantly B-cell lymphomas. You can see that at the very bottom is peripheral T-cell lymphoma, the most common type of aggressive T-cell lymphoma. And you can see that the 5-year survival is 20 to 25%. And that's significantly inferior even to some of the aggressive B-cell lymphomas. In the past, what we've done is we've treated T-cell lymphomas just like B-cell lymphomas. We've taken the aggressive lymphomas, lumped them all together, and given them aggressive chemotherapy. When we retrospectively look at how the T-cell patients do compared to the B-cell patients, this is data from the GILA, which is a large group in Europe, you can see that the patients with T-cell lymphomas did worse than patients with B-cell. These are all aggressive lymphomas. There's one subset of T-cell lymphoma, the anaplastic large cell lymphoma, that does better, but by and large, the rest of them fall down here in this curve. Well, what is T-cell lymphoma? T-cell lymphoma is actually many diseases and many clinical manifestations. And I promise you, this is the only clinical slide that I'll have in terms of showing you gross pictures. But these are the different kinds of manifestations of the disease in many different parts of the body. If you look at actually how the disease is classified in the WHO classification system, the best way we have to classify this group of diseases is based on how they present. So there's the nodal group, there's the cutaneous group, there's the so-called extranodal group, which includes things like enteropathy type T-cell lymphoma, hepatosplenic, and subcutaneous paniculitic. And then there's the leukemic group, which includes different types of leukemia, NK leukemias, and adult T-cell leukemia. The frequency of these different subtypes was determined based on a large retrospective study done around the world of 1,300 cases. And you can see that predominantly the nodal types, the peripheral T-cell lymphoma, angioimmunoblastic, comprise the larger number of cases. But you can also see, again, this is around the world, you can see distribution of several of the other types. And one of the things that I remember is that the ones that actually have the longest names are the ones that are the least frequent. Well, the prognosis, it turns out, is actually similar for many of these subtypes, despite the fact that they have different names and different frequencies. If you look at overall survival from that very same study of 1,300 patients, you can see that with the exception of these anaplastic large cell patients that do well, by and large, all of the other patients do not do well. And you can see that the median survival overall for this group of patients is 3 years or less. So from one point of view, it really doesn't matter actually what you call them, as long as they're not the anaplastic large cell type.
Dr. Foss, do you want to just point his back to that one slide? Most importantly, the best pitcher in the American League is a survivor of the first type of lymphoma.
Exactly. So if you have this type, you're okay.
If you're a pitcher for the Red Sox.
Right. And we know the Red Sox is the best team, and let's not talk about this year. Okay. Let's move on. In terms of aggressive approaches, these patients do poorly, so we need to treat them aggressively. And many strategies used upfront chemotherapy and then an autologous stem cell transplant. And that, in fact, is considered a standard in many parts of the world. But one of the problems is certainly if you can get transplanted, you do have a chance that you might do well. But many patients never make the transplant. So about 40% of patients treated with first-line aggressive therapy never go into remission, so they can never have a transplant. So clearly there's a large group of patients who need additional therapies. Now, in terms of understanding this disease, we have to look at what we can actually learn about the disease based on its natural history and whatever we understand about the biology. So if you look at the epidemiology of this disease, you can see some very interesting trends. If you look in North America, Europe, and Asia, you can see that the distribution of disease is different. In Asia, you can see a significant number of patients with adult T-cell leukemia, as well as these NK T-cell leukemias and lymphomas. Whereas in Europe and the United States, predominantly you see the peripheral T-cell lymphoma, the nodal diseases. And one other interesting thing is if you look at the enteropathy type, you can see that there's a higher frequency in Europe and the US compared, say, to the Far East. So this tells us a little bit about what perhaps is causing some of these diseases. And in fact, if you look at Japan and compare it to Western countries, in Western countries, 87% of lymphomas are B-cell, and as I mentioned, about 13% are T-cell. Once you get into Japan, you can see that about 22% are T-cell. And then if you move even further to certain areas of Japan in which HTLV1, say, is endemic, you can see that almost half of the T-cell lymphomas are accounted for perhaps by an association with HTLV1. So again, understanding a little bit about the epidemiology perhaps points us to etiologic factors in these patients. In fact, we know that HTLV1 is a causative factor, and HTLV1 is endemic not only in Japan, but also in the Caribbean basin. We also know that EBV is endemic in the Far East, and we know that EBV is a risk factor for some of these NK lymphomas, but also it's a risk factor for angioimmunoblastic as well as some peripheral T-cell lymphomas. And we also know, in terms of looking at that intestinal T-cell lymphoma, which was interesting that it was higher in Europe than in Asia, we know that disease is frequently associated with celiac disease. And of course, celiac disease is a disease of Caucasians, primarily Northern Europeans. Thinking about the immunology of this disease now, as I mentioned, these are many different diseases, and one way to think about them is to categorize them based on the cell of origin. And if you look at the immune system, there's two different arms of the immune system, the innate and the adaptive immune system. And this is just very brief and hopefully won't offend some of the immunobiologists in the audience. But my simplistic way of looking at it is that the innate immune system is really your first barrier. It does not require antigen sensitization. It's a primitive line of defense against pathogens and primarily exists at mucosal barriers. And so the kinds of T-cell lymphomas that arise in those sites, such as intestinal T-cell lymphoma, the gamma delta diseases of the skin, and some of the NK lymphomas of the nasal cavity really arise from this arm of the immune system. Whereas the adaptive immune system represents part of the immune system that has already seen antigen, that is antigen sensitized. And these types of lymphomas predominantly reside in lymph nodes, PTCL, angioimmunoblastic, and anaplastic large cell lymphoma. So people talk about immune dysregulation and how that may give rise to T-cell lymphoma. So what really is the evidence for this? I mentioned ongoing immune stimulation from viral infections such as HTLV1 and also EBV, and we know that that is associated with T-NK lymphomas. However, we don't know which is the chicken and which is the egg. We do see the association. We also know that chronic antigenic stimulation with impaired T-cell responses is a characteristic of celiac disease, and perhaps that is what leads to the development of T-cell lymphomas in these patients. And then finally, if we look at the situation of immune suppression, which we see in patients with solid organ transplantation as well as in Crohn's disease, we do see an increased frequency of certain types of T-cell lymphomas of that gamma delta type, again, the ones arising from the innate immune system. So I think there is evidence suggesting that at least in some of these patients with T-cell lymphoma, there is an element of immune dysregulation that leads to the development of these diseases. So then taking that one step forward, looking not only at the environment, but also looking at the host or the patient, there's been a significant amount of interest in looking at predisposition to developing these lymphomas. And this is actually work by Yaoi Zhang and Tongzheng Zhang at this institution who've collaborated with our group to do a SNP analysis of patients with T-cell lymphoma. This was actually part of their larger study, the InterLymp study, where they looked at patients with all types of lymphoma. But subsetting out the T-cell patients, we found that, in fact, there were certain SNPs that were strongly associated with the development of T-cell lymphoma. Again, this is a case-control study. And particularly, we're interested in chemokine and cytokine SNPs. And to take this the next step forward now, what we're trying to do is actually look at whether any of these specific SNPs are associated with particular outcomes and whether, in fact, we could document defects or alterations in functional immune status in these patients. So talking about all of these diseases, I've talked about a lot of different names here. And one of the questions that really comes up for those of us in this field is, are these diseases or are these syndromes? And what is the difference? So if you actually look at the definition of a disease is an acquired morbid change in any tissue by an organism with a characteristic symptom that's caused by a specific organism or alteration. And so say in the case of HTLV1, we know that there's a virus, that virus intercalates into DNA, and there's a specific association with the development of a disease, namely HTLV1-associated T-cell lymphoma. Also, with anaplastic large cell lymphoma, I'll talk about in a minute, there is a specific translocation. However, for many of these diseases that I'm talking about today, they're really syndromes. So they really are an aggregate of signs and symptoms associated with a morbid process and constituting together the picture of a disease. And a good example of that is the intestinal T-cell lymphoma. If it's T-cell and it occurs in the intestine, it must be intestinal T-cell lymphoma, right? Well, actually, that's wrong because NK lymphomas can present in the intestine, gamma delta T-cell lymphomas can present in the intestine. So really, even within this one group, there are multiple different diseases. So this is anaplastic T-cell lymphoma, which is a specific disease characterized by a translocation of chromosomes 2 and 5, creating a fusion protein. This disease can be advanced in many patients, and it's very common in children. The specific translocation is the NPM-ALK fusion protein, which is an oncogenic kinase, which activates JNK and Jun kinases and signaling pathways and leads to transformation of the cell. So here's an example of a specific chromosomal translocation leading to an oncogenic change that leads to a specific disease. However, in the case of paniculitic T-cell lymphoma, we have patients who present with these subcutaneous nodules. We do a biopsy, and we see that there are tumor cells surrounding the fat globules, and that is the definition of the disease. However, there's a significant amount of heterogeneity in terms of how patients do. And only recently we found that, in fact, there are two groups of patients. Patients who express the alpha-beta component of the T-cell receptor and those who express the gamma-delta component. And you need to do a special immunostain to determine which category your patient falls into. But clearly you can see that there's a very big difference in terms of overall survival between the two. Another way of looking at how to distinguish these diseases is again focusing back on the biology and looking at the distinct cell of origin and trying to figure out how to characterize them, because oftentimes they look very similar under the microscope, and there's oftentimes confusion on the part of the pathologist. So we need to learn more about what specific markers can distinguish the different categories. This is an example of PTCL and angioimmunoblastic T-cell lymphoma, and what we've learned is that if you look at antigen-presenting T-cells, they fall into the effector, the memory, central memory, or the effector memory categories based on the expression of CCR7. So if you just look at CCR7 staining, you can already identify angioimmunoblastic T-cell lymphoma as an effector memory type lymphoma versus peripheral T-cell lymphoma unspecified, which is a central memory type cell. So again, immunostaining will be very helpful, and also understanding where these cells come from will be helpful in terms of learning how to treat them better. Angioimmunoblastic T-cell lymphoma, I just want to show you this slide to tell you that, in fact, we treat peripheral T-cell lymphoma and angioimmunoblastic T-cell lymphoma with the same drugs exactly the same way. When you start looking at microarray signatures, you can see that they're clearly very, very different. There's clearly a chemokine-cytokine type signature in PTCL, and there's more of a B-cell signature in angioimmunoblastic T-cell lymphoma. So why are we taking these two diseases and treating them the same way? Clearly, we need to do better than that. Even if you look at the peripheral T-cell lymphoma unspecified, that one category, we know that by microarray that many of these cells have characteristics of activated T cells. And furthermore, we can divide them into CD4 and CD8 types. And then when we go further than that, we can actually see that there are 91 genes that are downregulated and 64 genes that are upregulated. So looking at what those genes are, you can see that they involve matrix, apoptosis, adhesion, proliferation, signal transduction, and transcription. And so now that we have a handle on what some of the pathways are that are being perturbed in these cells, perhaps we can use some of these to subgroup these patients again for prognostic and treatment purposes. And also perhaps we have a better understanding of this disease. I just want to say at this point that there is no animal model for this disease, and there are no long-term cell lines in culture to study. One easy way to identify these and look at prognosis is if you have an immunostain that picks out specific subtypes. And in this case, I mentioned chemokines, and it turns out that CXCR3 stains specifically for the AILT T-cell lymphomas, whereas CCR4 is more predominant in the ALK-positive mycosis fungoides and some PTCLs. So why is this important? Well, it's important because if you just use this one stain, you can actually show that there's clearly a difference in overall survival in patients who are either positive or negative, either for CCR4 or CXCR3. So adding one additional immunostain to your workup of these patients will allow you to identify, hopefully, the winners from the losers, and perhaps then you have a different treatment algorithm. Well, what about the conventional treatment? I told you that we treat everybody the same and everybody gets a CHOP type regimen. But if you look at a meta-analysis of a large number of patients, the 5-year overall survival is really very different for the different subtypes, again pointing to the fact that the biology is different. And the 5-year overall survival really isn't good for anybody. So in terms of where we are with therapy of these diseases, first of all, we really don't even know what the optimal first-line regimen is. I'm sparing you multiple slides that I could show you about other regimens that have been used, high-dose approaches, randomized studies that have been done in Europe, but none of those studies really show that any one combination regimen is better than another. We also really don't know what the role of consolidation therapy is with a transplant, because we know transplant can help some patients. We can't pick the winners from the losers yet. We also don't know whether we should be treating patients differently based on their histologic subtype. At this point, everybody gets the same treatment, but I've shown you lots of differences, and I think we need to start thinking outside the box and looking at the histologic subtype before we make a treatment decision. And finally, we know very little about allogeneic transplant. Well, I want to show you the NCCN guidelines for the treatment of relapsed or refractory peripheral T-cell lymphoma. The NCCN guidelines, as you know, are based on clinical evidence, and this is a recommended treatment algorithm. However, in the case of peripheral T-cell lymphoma, there really isn't much evidence. And so therefore, you can see that in the relapse setting, many of these patients get referred for clinical trials or even palliative therapy, because there really is no recommendation. If you can't get a response and move on to a transplant, there really is no kind of treatment algorithm here. So that's the state of the art as of right now. In terms of thinking about doing better, I showed you some data with looking at chemokine receptors. You can also look at various genes, and I think this is interesting data, just looking at NF-kappa B, whether it's positive or negative. That appears to predict for survival. So again, you could perhaps approach these two different groups of patients with different strategies. Other ways to approach this disease include looking at various surface antigen receptors and targeting them, looking at microenvironment factors such as angiogenesis, viral pathogens, or immune modulation, and looking at cell survival mechanisms and treatments that we have that might perturb some of these. I don't really want to talk about treatment today, but I'm going to just show you a laundry list of drugs that are out there. As of this point in time, there is no FDA-approved drug, but we just had an ODAC meeting, and this one drug, pralatrexate, got a positive vote, so that may turn out to be the first approved drug for aggressive T-cell lymphomas. In terms of new things, one new pathway that we're now focusing on is SYK, because SYK is traditionally a B-cell kinase, and we know that it's overexpressed in patients with peripheral T-cell lymphoma, but not on normal T-cells, and there are inhibitors of SYK that are in clinical trials. We have done some work with PDGF receptor. We know that PDGF receptor is overexpressed in peripheral T-cell lymphoma, and we do have drugs like sorafenib that can interfere with this pathway. And so we have a phase 1 trial looking at sorafenib in patients with aggressive T-cell lymphoma. But thinking to the future, we're actually looking at how to combine pathways. And so this is some data that was done by Marina in my lab, where she looked at combinations with sorafenib. And we found that there was an interesting and synergistic interaction with bortezomib. These are normal T cells, and this is the T-cell lymphoma cell line. And you can see that there's a significant interaction when you combine sorafenib with bortezomib. And so this would suggest that the next trial that we ought to do is this combination. So then in conclusion, as my time is running out now, what I think I've told you is that for the aggressive T-cell lymphomas, we really have an evidence deficit. The development of optimal treatments for these lymphomas really represents a clinical challenge due to the small numbers of patients, the lack of prospective trials, and the paucity of information relating to the biology. We have no trials to date that stratify patients based on biological features, and we do have some international efforts that are looking at trying to collect tissue. We have some novel agents that have been developed that are pathway specific and perhaps will overcome some of the limitations of conventional therapies, but we're still learning how to use these drugs. And we do have this international T-cell lymphoma project, which is involving many countries in the world, and Yale is a center for this. We're trying to prospectively collect tissues, analyze those tissues, and then look at outcomes to try to gain more information about these diseases. But ultimately, the only way we'll learn about these diseases is if the basic scientists and the clinicians get together and combine biology with new treatment approaches. And my final slide, I think, is just philosophical in terms of thinking about the mature T-cell lymphomas. The only source of knowledge is experience, and at Yale we are developing a tremendous experience with these patients. Thank you.